I will be talking in English because my Serbian is really bad, so I hope you don't mind. As Ivana said, I will be talking about bioprinting today, and also a little bit about how and what we do at IRNAS. So just to make a brief introduction of our organization, IRNAS is a non-profit institute working on the development and prototyping as well as manufacturing of complete solutions in technology ranging from electronics and mechanical engineering, software development, rapid prototyping, and also small to medium size serious manufacturing. So we really work in a, let's say, broad portfolio, and we try to work in projects which are oriented into different directions, ranging from wireless optical communication systems to IoT solutions, conservation technology, environmental monitoring for wildlife, as well as medical hardware, lab equipment, and also 3D bioprinting, which I will be talking more about today. So up until now, you should be fairly familiar with 3D printing in general. So how it works is basically you have a 3D object of which you make a 3D model. You cut it into slices, and then you build it slice by slice from bottom to the top again, and then you regain a physical 3D object. I think that was clear, more or less, right? So the principle of 3D bioprinting is relatively similar to that of conventional 3D printing, but with the addition of changing the material. So instead of using ABS or PLA plastics, or also other resins and similar materials, for bioprinting you use materials which are interesting for, let's say, biological or medical applications. There are different techniques for 3D bioprinting, similar also to conventional printing. So we have, let's say, inkjet-based printing, where you deposit small droplets in a series, or laser-assisted technologies like SLA or SLS, and also extrusion-based technologies, which is also my focus of work because it allows for the broadest spectrum of materials which can be used. So why is 3D bioprinting interesting at all? So there are several global challenges which we are now facing today, and this is a technology which has certain promises of aiding in the overcoming of these challenges. So one of the challenges is the need for transplantable organs and tissues for regenerative medicine. So just to give you a perspective on the current state, so in Europe, roughly 30,000 organs are transplanted each year, and about 60,000 people are put on waiting lists. So the demand is double that of the possible supply at the moment. And even the patients who receive, let's say, a donated organ, there's still a high chance that the body of the patient will reject that organ, which means that they are bound by immunosuppressive therapy, which really lowers their life quality in general. So it would be really interesting if it would be possible to make new organs basically from, let's say, patients' own cells to increase the biocompatibility and also to reduce the demand. So you wouldn't have to wait to be on the next in line to get actually a transplantable organ, but you could actually order one that is manufactured for you. And while this is really interesting and very promising to actually be able to make complete and complex organs, we are still a long way, we still have a long way to go. It is approximated that the first kidney that we will be able to transplant, which will be 3D fabricated, will come along in around 15 to 20 years. But even up to that point, tissue engineering and 3D bioprinting has various other applications which are useful for medicine already today. So even if we are able to make just simple tissues, these are interesting for, let's say, drug research and toxicology research, which can be done in the lab, but also in the pharmaceutical industry, for example, in a larger scale. So it replaces and reduces animal testing. It is much better than cell culture testing that was used as a standard before. And the other global challenge that is also related to the field of tissue engineering is livestock farming. So maybe you're familiar with this, but farming in general is one of the biggest environmental issues that we are facing today. From an environmental perspective, but also from an ethical perspective. We have a growing population, and also the standard of living in poorer countries is rising. Therefore, the consumption of meat especially and animal products is rising as well. So we are closely coming to the point where it is becoming unbearable to produce more livestock for food than it will be to sustain. So this is also something that tissue engineering could play a crucial role in overcoming these obstacles. So also just to give you a few numbers, so today the emissions of greenhouse gases from farming are larger than all the industry and transport that we have at the moment, and also 30% of all the drinking water that we consume in a year is 30% of it is consumed just by livestock farming. So this is something that also could be really interesting. And tissue engineering promises to find certain solutions for these problems. So if you could take, let's say, cells from a patient and grow a new organ to be transplanted or to be tested in a research lab, or again for livestock farming, to actually take out a piece of the animal and reproduce basically meat in vitro or in lab environment. So how do we get there? What are the requirements to be able to do that? I mean, if we are talking about it and we have the technologies for 3D printing and also for cell culture, extracting cells, why haven't we done it yet? So there's many different issues on a smaller scale that we have to go into, and each step you take makes the problem bigger and more complex. So the current, let's say, state of knowledge that we have in terms of requirements that will be necessary to fulfill, to make a new tissue, you first need source of cells. So these can be extracted either from the body directly in form of primary cells, so functional cells which are already differentiated. So if you wanted to have meat, you take muscle tissue and you cultivate it. There's also the possibility to take stem cells, so cells which are not differentiated yet but can be used and transformed during a process in the lab. Then you combine these cells with certain binding structures. So if you imagine bodies of animals and also plants and other organisms are not just connected to each other, but they also have certain space in between them, which is filled with proteins and other molecules, which we call the extracellular matrix. And this is something that we need to make or to combine with the cells that we are using to actually make the whole tissue. And then once we combine that, we need certain other influences and signals to stimulate the cells to grow and the tissue to develop, so such as growth factors, nutrients, and stuff like that. And 3D bioprinting is basically coming into play in this sector, so mimicking or making the extracellular matrix, or ECM because it's easier to pronounce, and trying to make scaffolds which will create the tissue backbone, or what we call scaffolds, on which cells can grow and tissues can develop. So we as a tech development organization, we are focusing especially on the fabrication and development of the fabrication process for 3D bioprinting. And what we see today a lot is that many researchers, especially in the medical field, but also in cellular agriculture, are working on the materials for the ECM to be useful for cells. So if you imagine you take a cell type and it has certain requirements, so also the material that you want to use needs to fulfill certain requirements for the cells to grow. And these vary among the cells. If you have different tissue types, different cell types, they have certain growth rates. You need to position them precisely. You need to have them attached to the scaffold, and you need to stimulate them to grow. And this is very complex in terms of material development. So a lot of research is now going into that field. And once the material is done, the fabrication process also needs to be done as well. So we now have a lot of, as the market, let's say, not just for conventional 3D printing, but also bioprinting grows, we have a lot of bioprinters that are already available on the market, which are mass-produced, but they only provide support for 3D printing certain material types. So what are some of the parameters that need to be fulfilled for material? So biocompatibility is the obvious one. So if you have a scaffold, it needs to be friendly to the cells, so the cells will attach, they will grow, they will differentiate. It needs to degrade. Ideally, it should degrade in a rate which is linear to the degradation, which is linear to growth of the cells, so the cells will replace it, ideally. It should have structural and mechanical properties which allow the structure to stay in the same shape or form during the process of growth. And also it should be printable. So this is often what we see this is difficult to combine. So these properties are so biocompatibility and degradation of the materials are often difficult to combine with printability as well. So this is where the process comes in. So to be able to fabricate the materials, you have to have precise resolution, and you have to precisely control the transition between solution and gel if you're using hydrogels, or also other mechanisms of making a material that is able to flow, so you can deposit it and then stay in place after it has been deposited. So I think this will be one of the biggest issues, and this is something that will require a lot of focus in the next few years. So combining the material development with the development of the technology in parallel to be able to make complex structures from custom-made biomaterials. And this is something that we are also working on at IRNAS. So just as an example, I would like to present a specific challenge which is also important in the field of tissue engineering today, and this is vascularization. So if you take a tissue, almost any kind of tissue that has high metabolic rates, let's say muscle tissue, let's say liver tissue, or kidneys, or heart, or brain, cells are very dense in such tissues, and they need a lot of nutrients, and they need a lot of oxygen, and they need to get rid of carbon dioxide, for example. And this requires a lot of blood vessels which are positioned very densely in the tissue. And it's very difficult today to actually fabricate such dense channel systems which are perfusable, but at the same time stable enough so they do not collapse because of all the empty spaces. And this is one of the biggest challenges that tissue engineering faces today. And in fact, NASA is co-sponsoring research prize to the first group that is able to make a centimeter high tissue that can survive for a month, and no one's been able to do it yet. And this is why we also haven't been able to make thick tissues in general. So we are working on the development of fabrication protocols as well, and this is something that we want to provide at least one way of going about it. So in addition to, let's say, normal scaffold building as you would expect it from conventional 3D printing, where you have a model with empty spaces, you just print the walls basically. This is already possible, but it has certain limitations in terms of the complexity of geometry, the minimum diameter of the channels that can be made, and this is not efficient enough for very complex tissues. So another possibility is also to make channels directly by combining a nozzle which can extrude, let's say, the inner material, which is like the medium, and then the outer wall of the channels themselves. And this is also very interesting at the moment, but it's difficult if you use this process to make connections between the channels, so you can only make long continuous ones. So this is why we tried also another approach that I just briefly want to show you. So this is 3D printing of channels in a substrate that you can use for tissue engineering, but then after curing, removing basically the channel system. And I would like to show you just a short video of the process of rinsing basically the channel system that we've been making. I don't want to go too much into the details of how this is made, but if you are interested in that kind of thing, I'll be happy to talk to you later on or tomorrow at the workshop. Yeah, so what you can see here is that the channels are perfusible. They can have complex geometries, are interconnected, and yeah, they are made from biocompatible hydrogels as well. So the next steps basically from here on is combining the 3D fabrication process with the cell culture process to actually make tissue engineering. So this is these are images from experiments that we've been doing with our partners, where we introduced blood vessel cells into the channels of the 3D printed structures, and they are green, which means that they are alive. They would be red if they would be dead. So this seems to be like an interesting approach. The next steps will be to make actual linings of the blood vessels and stimulate them to branch out and make smaller channels. So this is more or less all that I wanted to say today. If you have any questions, I'd be happy to answer them. Maybe a take-home message for bioprinting. It's 3D printing with biomaterials, which is useful for tissue engineering, and that will surely have an immense impact on global challenges we are facing today in medicine as well as in agriculture. And yeah, combining technology development with material development will also have an important role in getting to the next level. Okay. Really important to get functional vascularization to be able to make thick tissues, because yeah, this is the first limit. So just to give you an example, so every cell cannot be further away from the nearest blood vessel than, let's say, 100 microns. And yeah, this is very complicated to make at the moment. Yeah.
What's the thickness of your structure that you'll be able?
So you mean this one? This is about a centimeter high, but it's really a demo model. So Yeah, no, it's not. So here the channels are around 200 microns wide. We haven't yet tested how close you can position two channels, let's say. But this is also something that would be interesting to evaluate. Yeah?
And the printing, from the technical point of view, the printing was done in, let's say, one layer, like, or if there is like a different...
Yeah, you can make changes in the z-axis as well.
Maybe like 3D.
Yeah, so I didn't want to go too much into detail of the process of the fabrication, but since it's been brought up, so basically what we do here is we make microbeads from a hydrogel, and we make a very dense suspension of them. So basically, when you print with a needle, let's say, which is the nozzle in this case, you slide in between the particles. So the particles that are already all around, they hold the structure in place, so you can deposit the channel material, and you can go any form, basically, and then you cure the beads to cross-link them together, and then you rinse out the channel material. Size of the microbeads it varies a bit, so it's between 50 and 300 microns.
And the speed of printing?
I think here we were doing 500 millimeters per minute, something like that. Yeah. Yeah.
Just a question when it comes to using tissue engineering for that technique. Where exactly did you reach when it comes to, did you already manage to fabricate something, or is it something
I didn't hear you very
As you said that the thing that you have to do is to use tissue engineering to get food to meat.
Yeah.
And where exactly did you reach with that? Did you manage to fabricate something that's not eatable, or is it still just in process, just an idea that you might reach it?
So what we work on is the technology, the development of the technology and the fabrication process of the scaffolds. We don't actually work with the cells, so our partners work on the cell part. I have an image here. So the current state of the art for meat production, let's say, is like ground meat, basically. So this is 2015? 2013. So the first burgers that were made like this were like €250,000, and now the price has dropped dramatically. So I think now it's like 10 to 15 euros per burger, something like that. Supposedly the price will drop to 3, let's say, euros per burger in maybe till the end of this year or maybe next year. So this is this, but making steaks, for example, is not possible yet. So here what you really can do is grow cells loosely in a suspension, let's say, and then making muscle fibers in this case, and then you put them together. So it's not that you actually make an organized structure from ground up, basically. So you compact cells together.
If you do like some self-assembly, something like that, learn the cells to assemble in the right way to make some kind of scaffold, maybe it would be possible.
Yeah. So there are different actually approaches to that. So one of the research fields that's also emerging now is the fabrication of organoids. So you make small assemblies of cells, and then you cultivate them, and then they self-form certain structures. But it only works in a very small scale at the moment. So it's not like you can make the whole organ, but you make a small functional unit of that organ, which does the same function and is really interesting for research. But I'm not sure how it will translate into, let's say, organ transplantation. Yeah? Do you have a
I just wanted to say a couple of things because we had this workshop yesterday on cellular agriculture, which is actually tissue engineering for making food. And we were talking about, that's also part of the reason that Boštjan is here, because we're preparing together Institute Biosense and IRNAS and the company in Turkey that's called BIFTEC. So they work on making beef. And we talked about the general state of art at the moment, which is you can make something like this. And this took about 3 months and 3 very skilled technicians to work on, and huge amount of media and cells. So it was, that's why it was so expensive, €250,000. But now it's not necessary that you have so skilled technicians, but you still have to have a lot of media. And that's why it's still expensive. It's a problem of scaling up. So it's possible to make not only burger, but also some kind of sausage, something like that, but it's still expensive. So that's where we are at the moment. But then making steak, yeah, that will take a lot of time because you need the scaffold. And this for self-assembling, I mean, the cells first need to attach to something solid. So they start making their own matrix. But you can also print with these tissue, not organoids, but like assemblies of cells. And there is a Russian company, it's called 3D Bioprinting Solutions. And there was a speaker from there who spoke at FAB Belgrade 2018. And they print with these cell assemblies. So they already have many cells together. And they printed the trachea, which they implanted. So they work, they're quite advanced. So also with them we talked for some connections for the clean meat and everything. So yeah, it's ongoing. It's a pity that that workshop, that you didn't come for that, but we can organize it together, I mean, once again. Anyway, yeah, so...
there's also, just to go on the cells assembly, so there's also the same guys presented that at Belgrade. There's different approaches to 3D printing, or something that isn't really 3D printing anymore, but is controlled assembly of cells. So what they are doing is magnetic levitation, and also there's the possibility of acoustic levitation to bring like cells in place.
And this with magnetic printing, they are testing in the International Space Station.
Yeah, they just got all the permissions.
It's called Organoid, the 3D printer that's going up there. And yeah, you need magnetic because no gravity, so you need that. So, okay, so we should organize the whole conference on bioprinting, actually. I think that's the next step. Okay, so any more questions for Boštjan? You can also ask later today and tomorrow. Both lectures from this segment, Boštjan's and Sanja's now, also have like a sequel tomorrow in the form of workshops.
Yeah, a demo, let's say.
Demo workshop for you, but really practical one for microfluidics.
Yeah.
So thank you, Boštjan.
Yeah.
Oh.